Hi, I'm Dr. Evan Matthews. I'm here to talk about bioenergetics. So this is part 2 of a two-part video series on bioenergetics. So if you haven't watched the first part, please go back and watch that. That's going to talk about essentially where we get our energy as far as foodstuffs, so carbohydrates, fats, proteins, and it's going to go over a lot of the definitions that are going to be necessary for this part 2 video. So if you haven't watched that, go watch that first. I'll put the link in the description below. All right, so if we are looking at bioenergetics and we start to think about it with exercise, we find out that different pathways are the primary pathways depending on how intense the exercise is going to be and how long the exercise is going to last. All right, so a long, slow, less intense form of exercise is going to use primarily aerobic metabolism, and it's going to be using primarily fat instead of carbohydrates as its energy substrate, where a higher intensity exercise is going to be using primarily anaerobic energy sources, so much faster versions of ATP liberation. All right, so if we look here, we have our immediate energy sources. So we have something I call, just me personally, call stored ATP. All right, so what I mean by stored ATP, it's the ATP that's inside the muscle, just waiting to be used, that was produced a while ago. Last time you used the muscle, it was produced at the end, and it's just kind of sitting there and available to us. All right, so we do have a small amount of stored ATP in the muscle. It's not much. It can only last a second, 2, maybe 3 seconds of activity, but we do have some. All right, so first our body is going to go through that stored ATP before it starts to produce ATP, because it's essentially only going to produce ATP to replenish the ATP that was used up. All right, so we used up our stored ATP, and the first replenishment of ATP is going to come from the phosphocreatine system. All right, so phosphocreatine is very, very fast. After that, the next fastest system is going to be glycolysis, where we break down glucose into ATP. All right, so glycolysis is pretty fast, but not nearly as fast as phosphocreatine. All right, so glycolysis and phosphocreatine are our two anaerobic energy sources. When we start getting into aerobic metabolism, the end products of glycolysis can go into the Krebs cycle and the electron transport chain, which produce a lot more energy from those end products than it does through the anaerobic part, which is glycolysis. And you'll see that here in a minute. We also have the ability to use fats, so we can go through beta oxidation inside the Krebs cycle, inside the mitochondria, which releases something called acetyl-CoA, which you'll see in a minute, that goes into the Krebs cycle and then eventually the electron transport chain. So the Krebs cycle and the electron transport chain are going to be a common endpoint for all aerobic metabolism. All right, so we can also produce our own glucose, if you again go back to that first video, through a process called gluconeogenesis. So if we produce glucose, it's going to be used up in just the same way that glucose was used up when we consumed it. So it would go through glycolysis, and it can either end with glycolysis if it's anaerobic, or if it's going to go through aerobic pathways with the end product glycolysis, that pyruvate acid is the end product. It can then go into the Krebs cycle and electron transport chain. All right, so let's go over this in all this in more detail. The stored ATP that I mentioned, again, just ATP that's just sitting there, it's going to be able to fuel the body for about 1 to 3 seconds of exercise, and it's stored in the cytosol. All right, so just the fluid portion of the cell. All right, so this is going to go through something called ATP hydrolysis, which is where we break down ATP and releases energy for use. All right, so we have our ATP molecule here. We have ATPase, which is the enzyme that breaks it down. We have on the other side of the reaction, ADP. So ADP is 2 phosphates, where ATP was 3 phosphates, and so we split off an inorganic phosphate, and in the process we released energy. All right, that energy is then used to do some sort of work. Once we use up all the stored ATP, we then have to replenish it. Again, phosphocreatine is the first system that we use to do that. So phosphocreatine is also in the cytosol, and both of these require no oxygen in order to take place, so they are anaerobic. This is going to be the primary energy supplier for intense exercise lasting between 3 and 15 seconds, right? And basically what's happening here, it's a single step, so it's going to be very, very quick. So the more steps a process takes, the slower the process is. All right, so we have phosphocreatine here. It's going to break apart, and it's going to give one of its phosphate molecules to ADP and create the ATP in the creatine that's just by itself. All right, so that's how you split off the phosphate, you give it to the ADP, and create ATP again. All right, and the enzyme responsible for this is creatine kinase. All right, so once we burn through our phosphocreatine stores, we need to start using up glucose. All right, so it's going to we're going to go through the process called glycolysis. All right, so glycolysis can be split into two different segments. All right, so you can see there's a lot of stuff going on here, a lot of chemical reactions, a lot of steps. All right, but the first several steps of glycolysis are what we call the investment phase. So everything right now inside this purple box is the investment phase. We start with blood glucose. So we bring blood glucose into the cell, and in the process we actually use up one ATP molecule, and that once this process happens, we end up with something called glucose 6-phosphate. So once you have glucose 6-phosphate, it's stuck inside the cell, it's never going to leave the cell again. So this process here is essentially bringing it in, sticking it in the cell, and keeping it in the cell. A little side note here, glucose 6-phosphate is the starting place if you want to store glucose. So the storage form of glucose is muscle glycogen if it's in the muscle, or liver glycogen if it's in the liver. So if you have glucose 6-phosphate, you can bring it through this pathway here in order to store it as muscle glycogen, and then later on when you need that glucose, you simply break down the muscle glycogen through glycogenolysis, and you end up back with glucose 6-phosphate. So this is sort of an extra storage loop that can take place. All right, so anyways, going down this initial loop here, or initial path, again, blood glucose comes into the cell, uses up an ATP, becomes glucose 6-phosphate. That is then turned into fructose 6-phosphate, and then turned into fructose 1,6-biphosphate, or bisphosphate. So this is also an energy utilizing step. So we're going to use up another ATP in this step here. This is also where phosphofructokinase comes in, which is the enzyme which is the rate limiting step for glycolysis. Right, so this step here, though, producing the fructose 1,6-bisphosphate, is the end of the investment phase of glycolysis. All right, so in this process, we used up 2 ATP molecules, and we haven't produced anything. When we go to the harvesting phase, so now everything in this purple box is the harvest phase of glycolysis, we split the fructose 1,6-bisphosphate into 2 different identical molecules of glyceraldehyde 3-phosphate. All right, so once we have these 2 molecules, we have 2 separate paths. These 2 separate paths are going to do the exact same thing. So if one path produces an ATP molecule, so does the other path. So for every 1 ATP molecule that I mentioned, it's actually 2 ATP molecules being produced. All right, so but the first thing being produced here is an NADH, which is a high electron carrying molecule that's going to eventually be used by electron transport chain to produce ATP. All right, so but we made one in one side of the path, so we make another one in the other path. So again, we're making 2 NADHs here. Then we make then we make an ATP, but again that's 2 ATP, and then at the very end we make another ATP, which is again 2 ATP. So we have essentially 2 ATPs being used up in the investment phase up here. We have 4 ATP being produced that are down here in the harvest phase, and so we have a net of 2 ATPs that are going to come out of glycolysis. We also have two NADHs that are being produced in the investment phase, which again go eventually to become ATP, or to produce ATP. We also have these last two substrates here, the pyruvate molecules. So we have two pyruvate molecules being produced. All right, so those are important because those are eventually going to go into the Krebs cycle. I need to talk though about that for just a moment. So we produce these pyruvate molecules, and ideally our body wants to put it into this preferred path, which sends it to the aerobic metabolism side of things to be used up in the Krebs cycle and to produce eventually ATP. However, aerobic metabolism is very slow. There's a lot of steps to it. So if we are producing glycolysis, or if we are going through glycolysis too quickly, we are going to produce too much pyruvic acid for the aerobic metabolism inside the mitochondria to keep up. So if that happens and you completely fill this pathway, you're going to have some overflow pathway. So some of the pyruvic acid is not going to get into that pathway, and it's going to go a different pathway. That other pathway is going to eventually use lactate dehydrogenase in order to produce lactic acid. All right, so lactic acid is that thing that makes your muscles burn when you exercise at a high intensity. So it makes your muscles burn, you feel fatigued, you get that sort of hard, heavy feeling in your muscles. That is the lactic acid. So that means you're producing more pyruvic acid than what can go into your aerobic pathways, and so it's being turned into lactic acid just to basically deal with the excess pyruvate. All right, so in that process, you're also using up the NADH that was produced during glycolysis. So you're using up a pyruvate, you're using up an NADH molecule in order to take care of the pyruvic acid and turn it into lactic acid. So this is something that can be released into the bloodstream, can become glucose again through gluconeogenesis in the liver or even in the muscles, or it can sit in the muscle, and eventually if the rate of energy production slows down enough where aerobic metabolism catches back up, this lactic acid molecule can actually be reversed back into pyruvic acid. So this is a reversible reaction, and then it can go into the original pathway into aerobic metabolism. The key points here for glycolysis is it's going to be the primary energy producing pathway for exercise. That's 15 seconds to about 2 minutes of length. It's going to happen in the cytosol, and it's an anaerobic. Right? It's important to note that this is anaerobic. All right, so one thing I didn't completely mention that I kind of talked about, but so we have the blood glucose comes into the cell and in the process uses up one ATP molecule. So if you're starting with glucose that's in the blood that has to get into the cell, you're actually going to end up with two ATP molecules that are the net. But if you're starting with muscle glycogen, which is essentially blood glucose that got into the muscle a while ago and got turned into glycogen for storage, if you're starting with the glucose that's stored inside the muscle glycogen, you're going to end up with 3 ATP molecules from glycolysis. That's because you don't have to expend the extra investment molecule to get it into the cell, because you did that a long time ago. You also get 2 NADH molecules from glycolysis if it's being fed into the aerobic metabolism pathways. If it's not, those 2 NADH molecules and the 2 pyruvate molecules that also get produced get turned into lactic acid. So if your glycolysis is happening at a fast rate, so faster than what aerobic metabolism can keep up, you end up with 2 lactic acid, no NADH, and no pyruvic acid. If it happens slowly and aerobic metabolism can keep up, you end up with 2 pyruvic acid molecules that go into the next pathway. We're going to talk about the Krebs cycle, and you also have 2 NADH molecules that are going to go into the electron transport chain. So both of those are part of the aerobic energy pathways. So very different end products depending on if it's happening quickly or slowly and whether or not aerobic metabolism can keep up. Now we're going to go into the Krebs cycle. But before we can get into the actual cycle of it, so this circle loop here, we need to produce acetyl-CoA from that pyruvate molecule. All right, so the process of changing pyruvic acid into acetyl-CoA is called pyruvate decarboxylation. Right, so in that process, we create an NADH molecule, which is another one of those, again, high energy electron carrying molecules, and we also produce a CO2 molecule. So this is the first time we're seeing CO2 produced by any of the pathways. So this happens, we get acetyl-CoA, and that's going to go into this Krebs cycle, which just loops and loops and loops around. So it starts and ends with acetyl-CoA, and you have a lot of products that come off in that process. All right, so it's important to note that we made two pyruvic acid molecules in glycolysis, because remember, glycolysis is split into these two pathways, so you get two different pyruvic acid molecules, which means for every glucose molecule broken down, this Krebs cycle is going to cycle through 2 times. All right, so everything that is produced with one of these cycles is actually going to be produced twice. All right, so we're just going to go through, we're not going to go item by item here, but we'll talk about the key points. Right here we have an NADH produced, we also have a CO2 molecule produced, we have another NADH and another CO2 molecule produced in this step. Down here we make something that's called guanosine triphosphate, so GTP. Our bodies can, it's basically another high energy molecule, kind of like ATP, but our bodies don't really use it. It gets immediately broken down and gives us energy in phosphate off to an ADP molecule to produce ATP. Up here we have an FADH2 molecule being produced, so this is similar to an NADH where it's a high energy electron carrying molecule. We have some water coming into the system. We have another NADH molecule being produced right here. From the Krebs cycle, again, if we're talking about glucose being broken down, going through this Krebs cycle twice, so going through 2 cycles, you're going to end up with 2 ATP molecules that are produced here. You're going to end up with 8 NADH molecules that are produced 1, 2, 3, and 4. So do that twice, that's 8. And we're going to end up with 2 FADH2 molecules being produced right here. Right, so a lot of things being produced from the Krebs cycle. There is ATP, but most of what's coming out of it is going to be these high energy electron carrying molecules of NADH and FADH2. They're eventually going to go into the next pathway we're going to talk about, which is the electron transport chain. The Krebs cycle happened inside the mitochondria, so does the electron transport chain. All right, so we need to talk a little bit about the structure of mitochondria. So here's a sort of overview, or a big picture, I guess, of a mitochondria. You can see this outer brown membrane, then you have this inner membrane space, this yellow, and then we have an inner membrane. All right, so we have a 2 membrane organelle here with some space in between. That's really important because we're going to be using that space in order to create a gradient. Remember, anytime you have a gradient, you can use that gradient to do work. We have a number of different molecules here. Probably not important that you know all of them. I am going to talk about them briefly, but essentially we have these different complexes here. So we have 4 different complexes and some carrier molecules. All right, so complex 1 is going to break down NADH into NAD, so it's low energy form. In the process, it's going to pull off the high energy electron that I've been talking about. So it's going to pull that electron away from the NADH, and it's going to pass that on to coenzyme Q, which is a carrier molecule here. In this process, it's going to pump hydrogen into the inner membrane space. All right, so the coenzyme Q molecule is going to take this high energy electron and it's going to take it over to complex 3 here, which I'll get to in a second. Because between those we also have this sort of extra pathway, this complex 2 pathway, that's going to break down the FADH2 molecule into FAD, and in the process it's going to pull off that high energy electron, giving that also to coenzyme Q, and it's just going to get past to complex 3. So both complex 1 and complex 2 are going to be pulling high energy electrons off. Just complex 1 does it from NADH, complex 2 from FADH2, and they're both going to be giving those electrons to coenzyme Q in order to give it to the complex 3. Alright, so complex 3 is going to take that electron and it's going to hand it over to cytochrome C, which is another carrier molecule. In that process, it's also going to produce, or it's going to pump hydrogen into the inner membrane space. And notice how we're starting to get this buildup of hydrogen in the inner membrane space, and there's basically there's very little hydrogen in the inside of the mitochondria. All right, so cytochrome C then is going to carry this electron from complex 3 over to complex 4, where it's going to throw that electron onto a oxygen molecule, so an O2. This is the first time oxygen is being used anywhere in any of these pathways. And in the process, it's also going to use up 4 hydrogen ions. Right? So we're using up hydrogen ions, we're pumping hydrogen ions into the inner membrane space, and we're producing 2 water molecules in the process. All right, so important thing to note here, this oxygen molecule comes in at the very, very end of metabolism. It's the only place that it's going to come in, and all it's doing is it's accepting that that high energy electron that's getting passed from one complex to the next, and it's going to be using it to turn itself into water with some hydrogen ions. All right, so that is where oxygen gets used up. If this oxygen isn't there, it backs up all these other steps, and so none of those other steps happen. If those all back up, it backs up the Krebs cycle, so the Krebs cycle doesn't happen. And if that backs up, that means aerobic metabolism is slowed to a very slow rate or stopped. And so if you go back, remember, glycolysis is going to try to push pyruvate into the aerobic pathways, but if it can't, it's going to produce lactic acid. All right, so if oxygen isn't available at this very last step, it backs all those steps up, so you end up producing lactic acid. All right, so, but here, again, oxygen is the final electron acceptor in this pathway, and all these here are just, all this process happening in the electron transport chain is just trying to set up this hydrogen ion gradient. Again, so you have more ions on one side of the membrane than the other side of the membrane, so that's a gradient. So think of this like a dam on a river, right? So you put up the dam, you have a lot of water on one side, very little water on the other, and so that gradient from a lot of water to little can be used by some turbines in order to, it spins the turbines and it creates energy. Very similar thing is going to happen here. It has a lot of hydrogen here, very little hydrogen on the other side of the membrane, and this ATP synthase molecule is going to use that. It's going to bring the hydrogen in, it's going to literally turn like a turbine, and in the process creates ATP from ADP. So you're going to create energy using that gradient in that way. So we produce a lot of ATP in the electron transport chain, so 28 ATP molecules are produced from one glucose molecule that started in glycolysis. We also produce some water that's of note here, so we are going to produce 12 water molecules, and we're going to use up a lot of oxygen in the process. So again, this is where all the oxygen was used up. All the CO2 that was produced in the Krebs cycle. Aerobic metabolism is going to be primarily what we use during low intensity exercise, or just sort of getting around the house kind of stuff, or with very long duration exercise. So beyond 2 minutes, aerobic metabolism is the primary pathway for energy production. Real quick overview of glucose metabolism here. So we have glucose in the blood, so these are blood cells, and this is a capillary. It gets into the muscle cell and goes into glycolysis. In the process of glycolysis, it's going to produce 2 NADHs and 2 ATPs and 2 pyruvate molecules. And as long as there's enough, glycolysis is happening slow enough for aerobic metabolism to keep up, all this NADH and pyruvate is going to go into the mitochondria, where it's going to go into the, some of that, the pyruvate is going to go into the Krebs cycle as acetyl-CoA, going to produce a lot of NADHs and FADH2s and some ATPs and some CO2s in this pathway. Then these NADHs and FADH2s are going to go to the electron transport chain, where a lot of ATP in water is produced. So if we summarize this, we have 2 ATPs from glycolysis, we have none from the pyruvate decarboxylation that happened, which is just that turning of pyruvate into acetyl-CoA. But we do have some NADHs that happen there, that come out there. In the Krebs cycle, we have 2 ATP molecules 6 NADHs, 2 FADH2s. In the electron transport chain, we have 28 ATPs, and this is where all the NADHs and FADH2s are used up, so there's nothing being produced otherwise. All right, so a lot of energy from one glucose molecule. Alright, so fat metabolism is very, very similar. Fat metabolism is going to happen by bringing fatty acids inside of the muscle cell from the blood. In that process, it's going to be converted to activated fatty acids, and you're going to be using up ATP in that conversion. Alright, so that activated fatty acid is going to go into the mitochondria, where it's going to go down here through beta oxidation. Alright, so beta oxidation is going to go in circles, and it's going to keep breaking off pieces of the fatty acetyl-CoA that was produced by this process up here, and it's going to do that until there's nothing left. Alright, and it's going to be making acetyl-CoA from the fatty acetyl-CoA, and that's going to go into the Krebs cycle, just like the acetyl-CoA that came from glycolysis did. So at this point, it's identical. So you're just going to keep going through the Krebs cycle, producing these NADHs and FADH2s, and it's going to go into the electron transport chain and produce a ton of ATP, because there's a lot of energy inside of a fat molecule. All right, so generally speaking, we can get 3 to 4 times more energy from a single fat substrate than we can from one glucose molecule. So a lot more energy is in fat than what is in carbohydrate. We talked about enzymes a little bit in part 1 of this series on bioenergetics. We need to just briefly talk about the different rate-limiting enzymes here. So phosphocreatine, the rate-limiting enzyme, is the only enzyme. It's creatine kinase. Glycolysis, the rate-limiting enzyme, is phosphofructokinase. Krebs cycle, it's isocitrate dehydrogenase, and the electron transport chain, it's cytochrome oxidase. All right, so these are the rate-limiting enzymes for these different processes. What I mean by rate-limiting enzyme is these are the enzymes that are in highest demand and shortest supply. All right, so if you were able to build these up, so let's say you use exercise training and you train the person over time and they built more of these enzymes, they're going to have faster processes because they have more of these enzymes. All right, so again, they're the weakest link. So if you improve that weakest link, the whole process gets better and faster. All right, so these enzymes can be stimulated and they can be inhibited. Right? They're generally stimulated by the products of ATP utilization or breakdown. So these are all sort of breakdown products that suggest ATP is being used up. Right? They can, again, these enzymes can be inhibited by things that suggest that ATP is not being used up. So notice ATP is an inhibitor for all these processes. All right, so also these others that are listed here are things that are produced by these processes. So basically they feed back on themselves. If there's a lot of their end product around, they slow themselves down. If there's not a lot of end products, it's all being used up quickly, then they speed themselves up. All right, so that was a very surface level overview of bioenergetics. If you want a little bit more information, I would suggest getting a really nice bioenergetics textbook or an exercise physiology textbook. All right, so hope that was helpful. If you have any questions or comments, or you can just put those in the comment section below. Otherwise, please come back and watch another video. Thanks.